Now, a whole different organism we're going to attempt to isolate is something called Bacillus. Now, Bacillus is a gram-positive rod. It lives in the soil. It's everywhere, but predominantly it's in the soil. And so, if you want to get Bacillus, for instance, you don't do a rectal swab. You take a soil sample and you try to get it out. But think of all of the myriad of junk that's in soil. Think of all the organisms that are there. I mean, 1 gram of soil has like 10 to the 10th organisms in it. I mean, there's so many organisms out there, okay? How can you take some soil and just enrich for Bacillus, okay? And we're just using plain old TSA plates, okay? This is nothing special. These are just tryptic soil agar plates. How are you going to do it? Well, Bacillus is cool. Bacillus makes what are called endospores. Okay? An endospore is this little, very resistant, like little sac of material inside the cell that has all the cytoplasmic contents, all the ribosomes, all the DNA, and it's very resistant to heat and pressure and various things. So what we're going to do is take our soil sample and heat it up for about, I don't know, maybe 5 to 10 minutes at boiling. And what that will do is kill everybody in there, all the vegetative cells, okay, including vegetative bacilli. The difference is it won't harm the endospores. So we'll be able to take a sample out of there, put it on this TSA plate, and Bacillus is the only going to grow if we grow this aerobically. Okay? If we took this plate and incubated it anaerobically, the only thing you'd get would be Clostridium, because Clostridium makes endospores. It's a gram-positive rod, but it is a strict anaerobe. Okay? It's going to die in the presence of oxygen that we have here on the bench. Okay? So, how do you do this enrichment? And what we're going to do is we're going to take a no-heat sample, and then we're going to heat it up and put the heated sample over here, and we'll compare them and see what they look like a little bit later. So what do? You come over here, and you take a hot plate, let that thing come to boil, or close to it, and we're going to take our soil sample. We're going to just put some soil into a little test tube. This is just soil I got from SU, from the outside. Just take a little bit of soil, not much, okay, just a little, maybe a teaspoon. And do not worry about our fingers getting involved, because again, we're going to kill everybody except for the endospores, so it doesn't matter. We're going to put that in there like so. We're going to add a little water to this. Again, any kind of water will do. I happen to have some sterile water right here, and so I'm going to add a little bit of water to this, not much. I'm going to shake it up, shake it to distribute some of the cells that are in there. And now we're going to take a little bit of sample from this before we heat it. So I'll have a before heated sample. So let's go over and do that. So I got my soil no heat plate, and I'm just going to go ahead and just take a loop. So I'm just going to take some sample, put a little of the soil sample on there, maybe a little more. And it'll be fun to compare what grows here. Okay. Now I'm just going to streak these like I'll streak the others, and just go ahead, you remember, pull the loop, and we'll do tornado because we're just in a hurry. Haste makes waste, which is probably what I'll find out. Okay, There's our Bacillus, no heat. Now what we'll do is wait for this stuff to heat up. We'll pop our test tube in there and let it sit for about 10 minutes and let it go. And then we'll come back and do the very same thing. Streak it on that other plate that's heated. We'll incubate both of these at what temperature do you think? Okay, we're not going to be doing this at, it's not a body sample, it's from soil. So the rule of thumb is if it's something other than body, start at a lower temperature, start at 30 degrees C. So we're gonna incubate this at 30 degrees, okay? Why? Because there are actually lots of soil organisms that will die at 37. The organisms I work with called myxobacteria, they grow at 31 degrees. If you go to 33, 34, 35, they're dead. Okay? So if you're in doubt, a lower temperature is a better idea. They won't grow as fast, but start with a low temperature for environmental samples, lake samples, soil samples, rainwater, whatever, something from outside. Body, from the human body anyway, and probably those animals and things, 37 is the way to go. Okay? So I'll do this, turn this off because it's boring, and I will just go and incubate these. One thing I wanted to show you is when you're doing this, of course, you don't put a cap on the top of this, or you have a very loose fitting cap, otherwise stuff will explode. This is right now at 70 degrees C about, and I'm just going to let this sit here and have it hover between 70 and boiling. And you can see there's lots of bubbles down there. It's getting pretty dang hot. And again, all the vegetative cells, including the vegetative bacilli, are getting killed at this point. But the endospores will be unharmed, okay? So think about that the next time you're out backpacking and someone says, boil your water. Yeah, that'll get rid of a lot of stuff, but it won't do a darn to endospores, okay? They're the reason why you put the autoclave at high temperature and high pressure, because if you can, at high pressure, high temperature, endospores are killed. And that's how you rate autoclaves. You're checking to make sure you can actually kill off endospores, okay? Important to know. Just to prove to you that we're boiling away, this has been boiling for about 7 minutes. So what I'm gonna do is pull this out carefully. Turn this off so no one dies. And we're going to take this and we're going to streak it just like I did before. But this time, again, the sample has been heated. So I got my heated plate ready to go. Well, I got a whole tree that time. Put some sample on there. Put a little more on. More on. Get it? Speaking of which, it'd be nice if you could see what's going on. And then I just go ahead and streak this, just as we've done before. And I will go and incubate these guys, and we will see what our results are. Now I'm going to show you in a later video, because I haven't made up some of the materials for it, isolate Pseudomonas and how to isolate myxobacteria. Okay? Those are going to be sort of our last two that we're going to try to isolate. I might try to do Colobacter or a couple others because I got the time. And since you guys aren't going to get to do it, I want to show you how these things work. So what we'll do is watch these over the course of a few weeks or a few days, and I will give you updates as to what's happened and if they were successful or not. So there's staph, lactose fermenter that we hope is E. coli, and Bacillus enrichments. And what I want you to know about these is the plates, why they're useful. You should already know what a MAC plate's doing, why it is differential, why it is selective. If an MSA plate is differential or selective, is a TSA plate differential or selective, what it's doing, how we incubate these. So make sure you look at these videos and write down things like that so you can study and know the kinds of stuff I'll be asking you.